My name is Mireya Navarro, and I'm going to present my thesis project titled Biotransformation of Diosgenin using Laccases and Nanoenzymes. There is growing interest in developing plant-derived medicines that match or surpass the effectiveness of existing drugs, while minimizing side effects and offering cost-effective, environmentally friendly alternatives. Natural compounds have demonstrated in vitro and in vivo efficacy in treating numerous disorders, characterized by low toxicity, minimal adverse effects, novel chemical structures, and the ability to act on multiple targets, simultaneously modulating various molecular pathways and signaling mechanisms. Among these, steroidal sapogenins stand out as some of the most potent bioactive compounds from natural sources. Diosgenin, a steroidal sapogenin found in various medicinal plants, shares structural similarities with cholesterol and other steroids. These similarities to human hormones make it highly valuable, particularly as a precursor in the pharmaceutical synthesis of steroidal drugs. It also shows remarkable potential in treating a wide range of cellular and metabolic disorders, including Alzheimer's disease, leukemia, depression, thrombosis, atherosclerosis, cardiovascular diseases, and cancer. By its intrinsic medicinal properties that includes anti-inflammatory, antioxidant, anti-hyperlipidemic, hepatoprotective, antimicrobial, neuroprotective, and vasodilatory activities, among others. However, its high hydrophobicity limits its solubility in gastrointestinal fluids, reducing bioavailability and hindering absorption in the bloodstream, restricting its therapeutic application. This has driven the development of structurally modified derivatives to enhance its physicochemical and pharmacological properties. Laccases are multi-copper oxidases capable of oxidizing a wide variety of phenolic and non-phenolic compounds by the reduction of oxygen to water. Previous experience in the oxidation of structurally similar steroids and triterpenoids demonstrate that laccases, especially in combination with mediators, can modify hydrophobic centers and unsaturations present in the molecule. Their advantages include a broad substrate range and environmentally friendly reaction conditions, enabling the synthesis of novel derivatives with enhanced properties. These potentials stem from their ability to catalyze oxidative reactions without requiring harsh chemicals or extreme conditions, making them ideal for green chemistry applications where high selectivity is essential. Copper-containing nanoenzymes offer additional advantages, including a broader range of thermal and pH stability, reusability, and ease of production. Unlike Laccases, where the active site is located within the protein cavity, the catalytic centers of nanoenzymes exhibit a higher surface-to-volume ratio, making them more exposed and less sterically restricted. In the structure of Diosgenin, Laccases and nanoenzymes have the potential to epoxidize the double bond and oxidize the hydroxyl group. The objective of this work was to synthesize and characterize copper-containing nanoenzymes using FTR to analyze the functional groups, DLS to determine the particle size, and also enzymatic activity assays. The study aimed to compare the derivatives obtained from the biotransformation of Diosgenin using nanoenzymes and commercial traumatase versicolor laccases. Subsequently, the formation of any derivatives was assessed by monitoring enzymatic activity and performing TLC analysis. The copper-containing nanoenzymes were synthesized via a simple, non-toxic one-pot hydrothermal method using PMAA as both the carbon source and reducing agent. The biotransformation process involved the use of both free enzymes and lyophilized nanoenzymes. Diosgenin was dissolved in acetone, added to the corresponding buffer, and twin was used as a surfactant. The reaction kinetics were monitored at 1, 3, 6, and 24 hours by collecting 1 milliliter samples and measuring enzymatic activity using ABTS and PPD as substrates, as well as confirming TLC analysis. The laccase-like activity of the nanoenzyme was evaluated using PPD as calorimetric substrate. The increasing absorbance confirmed the catalytic activity, while subtraction of blank values corrected for non-enzymatic background. Result shows that catalytic performance was increased after lyophilization. Nanoenzyme site distribution shows a unimodal narrow peak with an average particle diameter around 30 nanometer, indicating uniform particle size. The narrow distribution and low polydispersity index indicate a relative homogeneous nanoparticle population and supports their potential in enzyme mimicking catalysis. The FDR spectrum of the copper nanoenzymes shows key functional groups directly related to their synthesis and catalytic properties. The peak of carbon double bond carbon stretching reflects the formation of aromatic or graphitic domains during the hydrothermal carbonization process. We have a band that corresponds to the symmetric stretching of carboxylate groups. These groups are crucial for coordinate copper ions. The peak of carbon-oxygen stretching vibrations may also participate in copper-oxygen coordination or stabilize the nanoparticles. Finally, the band assigned to copper-oxygen vibrations confirmed the successful incorporation of copper into the carbon framework or surface. Altogether, the spectrum proves that we obtain functional nanoenzymes with anchor copper centers, essential for the oxidase or Lacasse-like activity of the nanoenzymes. The biotransformation activity with nanoenzymes was monitored using controls without enzyme, without Diosgenin, and in water to rule out possible interference. The assay with one unit of enzyme showed a TLC profile very similar to the controls, indicating no detectable biotransformation under these conditions. Only the sample with 10 units displayed a slight change after 24 hours, which could suggest an initial indication of biotransformation. In the case of Laccases, when ABTS was used as a mediator, slight shifts were observed at 6 and 24 hours. The synthesized nanoenzymes exhibit uniform size and functional groups consistent with copper-oxygen coordination, supporting their potential for biotransformation processes and Laccases mimicking activities. Enzymatic activity assays confirm the catalytic performance of both Laccases and nanoenzymes. Preliminary observations of slight variations in TLC may indicate biotransformation, especially in the case of Laccases, where rotation times should be determined. In addition, HPLC analysis is being incorporated to confirm and validate this biotransformation. This work represents an initial step toward exploring the potential of Diosgenin for the development of new drugs.